in as well. And so it is my pleasure, he doesn't need much introduction at this point, but Dan Baruch, who's our director of the Center for Virology and Vaccine Research here, a professor at Harvard Medical School, and clearly a leading expert for which we are all incredibly grateful on the vaccine development as we see it today. So Dan, I think we've made you co-host, and we'll turn this over to you.
Okay, thank you very much, and good afternoon, everybody. It's a pleasure to see so many of you today. Let me try to share my screen. Okay. Can you see my first slide and can you hear me okay?
Yep, we're good.
Okay, great. So I was asked to speak for about 15 minutes on COVID-19 vaccines. Where are we? And in particular, I'll make comments about the Pfizer and Moderna vaccine products, which are going to be the first ones available, potentially even in the next few weeks. And give you my own perspective of essentially what we know and what we don't know about them. So first, I think the topic of COVID-19 really needs no introduction at all, given how profoundly it has impacted all of our lives, with well over 50 million global cases and well over 1 million deaths. And unfortunately, our country has borne the brunt of it. So we clearly need a COVID-19 vaccine for the primary purpose of ending this pandemic, but it also could be important to control future outbreaks. And regardless, vaccine development, regardless of the clinical need, vaccine development is an important contribution to science in terms of our understanding of immune correlates of protection. So as a brief background to the overall vaccine field, there's over 200 vaccine candidates under development, according to the WHO, which is a truly astonishing response of the scientific and medical community to this pressing global pandemic. And these vaccine candidates really comprise essentially all current vaccine technologies, including DNA vaccines, RNA vaccines, purified protein vaccines with adjuvants, viral vector-based vaccines, inactivated virus vaccines, and others. The first vaccines to show clinical efficacy, which we first heard about 2 weeks ago today, were the RNA vaccines, first from Pfizer and then Moderna, which is a new vaccine technology that's never been used before in a clinically approved vaccine. But the timeline to show clinical efficacy is actually not the most relevant timeline for ending the pandemic. The most important timeline is the timeline to produce and deploy billions of doses of vaccine worldwide. But given the events of the last 2 weeks, then I think we can say that vaccines will likely become available for emergency use authorization this winter, starting potentially in mid to late December, and then more broadly to the general population by springtime. So I imagine everyone heard this truly stunning news exactly 2 weeks ago on Monday, November 9th, when Pfizer was the first to announce the results of a phase 3 COVID-19 vaccine study with their RNA vaccine. The vaccine not only worked, but had a truly astonishing, it was reported over 90%, then clarified to be 95% efficacy, which is much higher than I think any of us anticipated, particularly for a respiratory virus, and would place these vaccines amongst the types of vaccines that have become the stalwarts of medical practice. Just a week later, also on a Monday, all these announcements seem to occur on Mondays, was the announcement of the Moderna RNA vaccine, which is constructed very, very similarly, only subtle differences in the lipid compositions between the Moderna and Pfizer vaccine. And similarly, Moderna announced that their RNA vaccine also had about a 95% efficacy. So very similar results from a very completely different company. And literally just today, was the announcement of the AstraZeneca vaccine, that this vaccine also showed substantial efficacy at preventing COVID-19 infection. This is a different vaccine. This is a chimp adeno vaccine, and had some curious results for which I think we don't have the full knowledge yet, but appear to be more effective in one dose formulation than another. But regardless, we now have 3 companies that have announced successful vaccines. And I think it's almost curious that these have been 3 past weeks, Monday mornings. But the field is actually larger. There's probably 10 to 12 vaccines in phase 3 clinical trials throughout the world, and probably 6 vaccines are going to be tested in the United States. The first 2 are the 2 I told you about, the RNA vaccines from Moderna and Pfizer. These vaccine studies started in late July and just announced efficacy in the last 2 weeks. The next vaccine was the AstraZeneca vaccine with a chimp ad ox vector. The UK studies started in the summertime, and it was the UK and Brazil studies that were read out today. The US study started in August, and that study will likely read out in December. The fourth vaccine that started efficacy trials in the US was the vaccine that we helped create with Johnson & Johnson, the Ad26 vaccine. And this phase 3 study launched in September, and hopefully we'll see efficacy results in December or January. And the next 2 vaccines are protein-based vaccines from Novavax and Sanofi, and these studies will likely start in December and then will be complete their trials sometime in 2021. So the question is how to accelerate vaccine development, which typically takes many years or decades. So the development of a COVID-19 vaccine in 1 year is truly unprecedented. And it's important even for those of you who are not intimately involved in vaccine development, because the question of vaccine safety is on everyone's minds. I'm sure it's on your minds, but it's also on the minds of the general public. And we have to be able to reassure people of the safety of these vaccines. Otherwise, the vaccine campaigns to stop the pandemic will not be successful. So the key aspects of acceleration of vaccine development is to use vaccine platforms that have been tested for other pathogens. So the process of entering clinical trials is faster than otherwise. And the development activities are typically performed in parallel rather than in series. For example, many companies have been mass producing their vaccines even during the course of the clinical trials rather than after they see the efficacy result. So upon approval, then the vaccines can start to be distributed almost instantaneously. This has required a substantial tolerance of programmatic and financial risk on the part of the companies, obviously supported by the US taxpayer. But the critical thing is that it's important not to compromise patient safety or regulatory integrity. The vaccines also have to be able to be mass produced and globally distributed in order to achieve herd immunity to end the pandemic. So for the Moderna and Pfizer vaccines, the really question is, what do we know and what we don't know about them? And so I've divided this into a few slides on the different topics that I think are most relevant. In terms of the products, they're very similar products. They're RNA vaccines encapsulated in lipid nanoparticles. Pfizer filed for FDA emergency use authorization last Friday. The FDA VRBPAC committee, I believe, is planned to meet December 8th through 10th. And so assuming they give it a positive review, then this vaccine, if approved by the FDA for emergency use authorization, could be expected to have an initial rollout to healthcare workers in mid to late December. Moderna is just a couple weeks behind and is expected to file for FDA emergency use authorization in the next couple weeks. And therefore, this vaccine could potentially be available for rollout to healthcare workers in late December and early January. The reason why there's a slight delay is because the FDA has required 2 months of safety follow-up of at least 50% of the vaccinees prior to reviewing an emergency use authorization package. So these are sort of the timelines of when these vaccines might be available to healthcare workers and first responders. So in terms of efficacy, the efficacy of both of these vaccines was truly outstanding, essentially 95% from Pfizer and 94.5% from Moderna. These are 2-shot vaccines at a schedule of week 0, week 3 for Pfizer, and a week 0 and week 4 for Moderna. The responses after the initial prime immunization were largely undetectable or very low, and the responses came up substantially after the second shot. So I would assume that I think it would be good to assume that you're not protected until a couple weeks or until a week after your second shot of the vaccine. Also, the question is, what does this efficacy mean? And the efficacy essentially reflects the case definition of COVID-19 used in the clinical trials. So the efficacy reflects the prevention of symptomatic COVID infection, including both mild and severe disease. No data is currently available for the prevention of asymptomatic COVID infection or for blocking transmission. And there'd be theoretical reasons to think that it's easier to protect against severe or symptomatic disease than asymptomatic disease. So we simply have no information yet on whether these vaccines or what the efficacy of these vaccines are in preventing infection. What we know is that they have a very good efficacy of preventing symptomatic infection. Also, we have no data whatsoever on the durability of protection because the trials are so big, they read out almost instantaneously. And so we have no idea if these vaccines are going to be effective for a very, very brief period of time, or if they will be effective for a prolonged period of time. We just don't know that yet. So safety is the most important part about any vaccine. And I would say that that includes vaccines developed in pandemic settings, because if there's not public trust, then the vaccines will fail. So a vaccine not only has to be safe and effective, a vaccine has to be safe, effective, and trusted in order to achieve enough population coverage to achieve herd immunity. So Pfizer and Moderna appear to have fairly good short-term safety. I say anticipated acceptable short-term safety just because none of us have reviewed the primary data yet. But assuming that the FDA reviews and then the general community reviews don't reveal any concerns, then there is acceptable short-term safety of these vaccines. What does that mean? It means that there is safety known for about 2 months. We also know that the reactogenicity of these RNA vaccines, for reasons that are not yet clear, is relatively low after the first shot and substantially higher after the second shot of the vaccine. And what the underlying immunology of that is not yet known. I think it's important to realize that there's no safety data at all for long-term safety of these RNA vaccines or any RNA vaccines, because there has never been an RNA product on the market for any disease. So the short-term safety looks good. There's no long-term safety, and there's no experience with any other RNA vaccines or any other RNA products. But the long-term safety will be monitored post-approval. So how about storage and distribution? Again, has been in the news a lot about these vaccines. Being a new technology, these vaccines do require a cold chain, and therefore distribution may be challenging, particularly for some certain areas in the US, probably not for urban areas like cities. So I don't imagine this will be an issue affecting Boston. But for certain areas in the US and for sure for much of the developing world, distribution of these vaccines will be a challenge, particularly for the Pfizer vaccine, which requires an ultra low temperature cold chain at -80 degrees. And the reason is because RNA is intrinsically an unstable molecule, and it's been made more stable through base pair modifications and through lipid nanoparticles, but still, it requires a substantial deep freezing. And no vaccine has ever required these sort of temperature requirements. And there's sort of innovative solutions of little capsules filled with dry ice that are going to be shipped to various places. In cities, it's probably not going to be an issue because there could be central storage facilities in major teaching hospitals or major cities. But in some areas, it will be much more of a challenge. And trying to get this vaccine to certain areas of the developing world will probably be very challenging, to say the least. The Moderna vaccine is a bit more forgiving in that it appears only to require a regular freezer for a frozen cold chain at -20 degrees Celsius. And also it's stable for 1 month at 4 degrees, which adds a lot of wiggle room. The next vaccines that may be approved would be the AstraZeneca and J&J adenovirus vector-based vaccines. And these would have a substantial advantage in that they don't require a frozen cold chain. So the rollout plan for a vaccine, the initial population is planned to be healthcare workers and first responders, and that will happen immediately. And then gradually, as vaccine becomes available, a rollout to the general population. First, elderly individuals with comorbidities, and then sequentially to other groups, essentially in terms of decreasing risk. So the last people to get vaccinated will be young, healthy adults. The question of pediatric vaccination is another question that currently there's no specific plans for. The rollout plan for adults is expected to follow recommendations provided by the National Academy of Medicine, Science, and Engineering. As far as I know, those are the recommendations that will be followed. The major issue that we will encounter now that it appears that we will have not one, but at least several COVID-19 vaccines available is going to be the limited initial COVID-19 vaccine availability. So initially will be the Pfizer vaccine, probably very quickly added on the Moderna vaccine. With these two vaccines alone, there would be an expected 40 million doses available by January, enough to inoculate 20 million people, which is still a pretty small slice of the population. Over the course of the spring, sort of February through June, there will likely be a couple 100 million doses available. And by the end of 2021, there could be 1 to 2 billion doses available for these two vaccines. The vaccine rollout in the US and the world obviously would be greatly accelerated if other vaccines also become successful. In particular, the AstraZeneca and J&J vaccines would be the next ones up for sort of reading out. The first AZ results came out today. Future AZ results will probably come out in the next month or so. So if these two vaccines would also be approved, then there could be a much more rapid and robust rollout of these vaccines to the US population as well as to the global population. These four companies have committed to at least 1 billion doses of vaccine each. AstraZeneca probably even 2 billion doses. So if there can be 4 or more vaccines eventually available for the world, then we might come close to covering the global population. So this is my last slide. The vaccine rollout in the US will be coordinated by the US government as part of the Operation Warp Speed program. 2 vaccines will certainly be a great start, but insufficient for a rapid US rollout. If it's just these 2 vaccines, it will be a relatively slow rollout over the course of 2021. And clearly will be vastly insufficient for the global population. So the global population is 7.8 billion people. So for a 2-shot vaccine regimen, that means we will need over 15 billion doses of vaccine, fewer if there are some single-shot vaccine regimens available. And so we need actually multiple vaccines to be successful to end the pandemic. And also, I think for yourselves as well as for all your patients, it's important to let people know that over the course of the rollout of the vaccine, it's still going to be critically important to follow all the public health guidelines of mask wearing, social distancing, and everything else you've been hearing about, because there will be a substantial period of time before vaccines will be able to control the pandemic. But I think we're in a position now that we are much, much more optimistic than we were 2 weeks and 1 day ago, in that the immediate future for us as a country in the next 2 to 3 months is incredibly bleak. But the medium-term future of what's going to happen in 2021, I think there is good reason for optimism. And I think there's optimism that science will ultimately triumph and will defeat this pandemic. I'll stop there. And if there's time for questions, I'm happy to take any questions.
Yeah, that was just great. And yeah, I mean, it's just perfect timing, I think, as we sort of face what the next, this next wave is going to bring to us to have at least some hope on the horizon that it's not forever. There's 3 sort of biology virus questions. So let me start with those and then I'll turn it over to Tony, if you don't mind, Tony. The first is, as you can imagine, is the question of for the mRNA vaccines, since there never have been in approved vaccines for humans, are there any theoretical side effects that you're worried about? People ask specifically about immunomodulation and heme malignancy, but anything else that you're thinking of?
Well, I think that there is just a lot of unknowns. And unknowns doesn't mean that anything is worrisome or bad. It's just that we don't know. So there's never been an RNA vaccine product that's been approved, and there's never been an RNA therapeutic product for other indications also approved. Based on the experience with the COVID-19 vaccine, I imagine there will be a lot more RNA therapeutics moving forward. I think that what we do know is that there is more reactogenicity with the second dose than the first dose. So there is some potentiation of reactogenicity. That was all still short-term and relatively well tolerated. But that's an area that we don't really know about yet as to what caused it. And then, of course, if there is some severe side effect that cannot be picked up in a large but short-term study, then of course that won't be detectable until the broader rollout occurs. But I will say that I think that the efficacy of these vaccines is really nothing short of amazing. Any sort of concerns have to be weighed against the very real ongoing transmission of the virus and the rapidly escalating death rate that we have seen in this country.
Yeah, and I will say as a clinical trialist, and I'm hearing that the placebo patients in the trials get the vaccine after. So we should have 80,000 people vaccinated for at least short-term safety data in a pretty short period, which is, if you sort of combine it as a platform technology approach, seems like a pretty robust initial sample size.
So many vaccines, just for comparison, many vaccines are approved based on a lot few person years. So these trials are huge. These trials are 30 to 60,000 people. So even if people are followed for 2 to 6 months, then eventually they will be followed for 2 years. But many vaccines are approved based on a substantially smaller safety data set if you think about person years as the element of interest. So the only issue that we don't have yet, and it's impossible because it hasn't been around long enough, is we don't have any long-term follow. So we have zero information of what the six month, 1 year, 2 year type issues might be.
So the difference in the cold storage obviously will lead to a lot of logistic differences. Is there a biologic difference about why their storage needs are different?
So I was actually asking one of my colleagues at Moderna about that. And the thought is that the companies have poured a lot of development time and expertise and investment into trying to tweak the lipid nanoparticles to make the particles more stable. So Moderna has invested in the development of different types of nanoparticles. And at least it is their belief that the specific formulation, the specific lipids, percent composition of the lipids helps to increase the stability of the RNA. I think, I'm not sure that's been externally verified, but it appears that the specifics of how the RNA is encapsulated might have an impact on the stability of the product. But it's an area that will likely continue to improve on. So I wouldn't be surprised if there is a huge investment over the coming years in making RNA therapeutics more practicable.
Yeah. I've managed to consolidate the questions at the moment into sort of 4. They're mostly clinical vaccine, I would say, speculation, because I imagine the data is a little too early to opine on. But Dan, we'll let you add them anyway. The first is, if you already had COVID or you have been vaccinated partially with one vaccine, should you get another? Would you get another? How would that pan out?
Well, so first, I think I should say that the data for these first 3 vaccines, Pfizer, Moderna, and AZ, have not yet been made public. So I think that we all, as physicians and scientists, we want to see the primary data. So that will need to wait. So we don't know a lot of the primary data yet. But the question as to, well, what happens if you were infected? Then can you still get the vaccine, or should you get the vaccine? The short answer is we don't know for sure. That's sort of the formal answer because it hasn't been tested in that setting. But what I do know is that some of the vaccines, including the J&J vaccine, does not use serology as an exclusion criteria. So there are individuals in the clinical trials that are seropositive at baseline that presumably reflects past infection. And in those, at least in the clinical trials that we've done here at the BIDMC, those individuals have some of the strongest responses post-vaccination. So it appears that, and this is just based on sort of a few cases, but those who have antibodies at the start, presumably reflecting prior infection, appear to have some of the most strong, the most robust response to the vaccine, the first shot, because then it's essentially the vaccine is a booster dose up front as opposed to a prime. But the formal answer, it hasn't been really tested in that setting, so it's difficult to give any definitive clinical guidance.
Yep, just absolutely what I expected, but very eloquently put. In terms, the other questions are about immune reactivity to the vaccine, since we know that the immune system rages out of control to bad effect. And maybe this question about anaphylaxis that Tony raises. How concerned are you about those consequences?
So I think that it's more precautionary. I think that the incidence, I don't know the numbers exactly because I don't have inside knowledge into the safety databases into those vaccines. But my understanding is that the incidence of immediate severe reactions is very, very low. So, I mean, I think that with any vaccine, including the flu vaccine, then people can have reactions, vasovagal, hypersensitivity, anaphylaxis. But I haven't heard of that being a substantial problem with any of the vaccines that have been developed.
Yeah, and it feels to me, again, it's sort of a clinical trials construct that there's an observation time, and it may be just the emergency authorization that they're kind of carrying it into that phase. That's purely my speculation, but it's not an unstandard thing to see after injections of any kind. How about a question kind of following up on your data slide where you said that we understand that we can prevent symptomatic transmission or symptoms from presenting, but not necessarily asymptomatic. And the question is there in the history of vaccines, a history of vaccines that didn't do both at the same time?
Yes, absolutely. I think that prevention of disease and prevention of infection are completely different. They're conceptually different and they're practically different. It's probably a lot harder to protect against infection than disease. And that's why we've seen that in all the trials reported, Pfizer, Moderna, and AZ, if you sort of read down into the bottom paragraph of the press release, then they usually make comments about severe disease. And the severe disease cases have skewed very, very strongly to the placebo group. So also in animal models, then we see a similar phenomenon that it's probably much easier to prevent severe disease than to prevent mild disease or asymptomatic disease. So we don't know. We have no knowledge. We have no data on the ability of these vaccines to block infection. What we know is that they can block symptomatic infection, which is most important for the individual, but obviously the purpose of vaccines is twofold. One is to protect the individual. The second is to provide herd immunity to block transmission of the population. So the latter is yet to be determined. I mean, let me give you an example. Many or maybe all of you have had the measles vaccine. If you get exposed to measles, then you very well might get infected, but you'll probably have a subclinical viremia and have anamnestic immune responses that will eliminate it before you become symptomatic. For COVID-19, for a respiratory virus, the consequence is that the theoretical consequence is that you still could be contagious. So if you get the vaccine, then since we have no knowledge about it, even if you're protected against symptomatic disease fairly well, at least for a short period of time, then as of now, we don't have the data to say that you are protected full stop, and you then you also don't have to worry about transmitting it to others. So that's why, at least for the early stages of the vaccine rollout, until we know more information, then we have to emphasize both for ourselves as physicians and to our patients that just because you got the vaccine doesn't mean that you can stop doing the other aspects of public health, such as mask wearing, social distancing, et cetera.
People are not going to be so happy with that message, but I get it.
But as the vaccine rollout occurs, if it's successful,
it'll get better and better. Yeah.
And if in 2021 we can get the majority of the population vaccinated with a combination of these vaccines, then hopefully gradually that will go away. Yeah. But I think if people think that once you get the vaccine you're protected, you don't have to worry about it, then I think that is an incorrect message to give.
So two more questions, if you don't mind. One is, what are the advantages of the J&J vaccine? Obviously, the refrigeration is an advantage. The technology is a little older, so probably a little more worked out, I would think. But any particular other thoughts?
So, well, first, The disadvantage is that that's not available next month. So, but the trials are ongoing. The phase 3 trials are enrolling very rapidly. In fact, they're enrolling at light speed. We're enrolling about 2,000 people a day. So the entire trial will probably be enrolled by December, end of December, and hopefully will read out in January. And J&J is manufacturing the vaccine just like Pfizer and Moderna and AZ. So if the trial is successful, of course, that's still an if at this point, but if it's successful, then there will be 1 billion doses of the J&J vaccine to add in to the other 3 companies. In terms of the vaccine itself, the J&J vaccine is being tested. It's the only vaccine that's being tested as a single dose vaccine. So it's also being tested as a 2-dose vaccine, almost as a backup. But if the single dose vaccine is successful, then that will have tremendous practical and public health advantages over the other 2-shot vaccines, because we know in any vaccination campaign, there's a significant drop-off of people coming back for the second shot. And that's true in the US, and it's even more true in the developing world. So that's one advantage. The second advantage is the lack of the need of a cold chain. And the third advantage is probably a safety advantage in that it's been around in different clinical trials for 10 years, and there's a licensed product on the market already, which is the I-26 Ebola vaccine in Europe. So it has a lot more longer-term safety data than the RNA vaccines. But as I said, the disadvantage is that it's not available yet.
All right, one more for you, and then I have one for Tony. What about immunocompromised populations? And I imagine the type of vaccine might matter a little bit here too.
So the good news is that all of the vaccines that we've been talking about today, first the RNA vaccines, then the replication incompetent advectors, and then the protein vaccines, all of the vaccines that are under study and will become available in the United States are not based on the virus itself. So there is no replicating virus vaccine that is being developed in the United States for COVID-19. So in that sense, then theoretically, all of these vaccines are likely going to be safe in immunocompromised individuals. The question is about immunogenicity and will they still work? And that's an unknown question, because for most of the phase 3 studies, people who are under immunomodulator therapy for rheumatic diseases or in immunosuppressive therapy for a variety of diseases were excluded. So we actually have very, very little data on whether the vaccines are immunogenic, and no data about whether the vaccines are effective in people with compromised immune systems, either intrinsically compromised or pharmacologically compromised.
And Tony, question.